Gene expression. So how genes are expressed means how do we take this genetic code and translate it into proteins, which ultimately determines our traits and characteristics. So the genetic information moves from DNA to RNA to protein. Now, all cells have the same type and number of DNA. So if we're talking about somatic cells, which are body cells, we're talking about 46 chromosomes. If we're talking about gametes, which are sex cells, we're talking about 23. So how can we have the same 46 chromosomes in every cell, but we have different types of cells that have different shapes, they have different sizes, and they do different things? Is that not all genes are expressed in every cell. So a bone cell is going to be different from a nerve cell based upon the genes that are switched on or switched off. And that's what expressed means. Which one of those genes will ultimately become protein that determines our traits, our characteristics. Mutations. So we have a couple of types of mutations that we'll look at here. Point mutations. Simple point mutations. Are going to probably affect one amino acid, and these are caused by substitutions. So if you look at this example, here's our DNA. This mutation probably is going to occur during replication during the S phase of interphase, and for some reason our G was switched out for a C. So what is probably going to be affected here would be a single amino acid, maybe. Now, not all of these substitutions are going to be detected. Sometimes the substitution results in the same amino acid, which is going to make the protein normal. Sometimes the substitution might change it to a different amino acid, but that amino acid is pretty similar to the original. So you might have a faulty protein, or you could have one that results in a stop codon and then your protein is incomplete. Now, frame shift mutations are going to shift the whole entire reading frame of your genetic sentence. Remember, our genetic sentences, we're looking at one side of DNA or the other side of DNA, and we read in sets of 3. So we read just like our codons. That's how we determine our amino acids. So a frame shift mutation is either going to be an insertion or deletion that's going to alter this reading frame, which includes 3-letter genetic words that happen to code for amino acids. So we could see with an insertion, what it's going to do is going to change our number of bases. We're going to have an extra, and a deletion, we're going to have one less. So you can see with this insertion mutation what it looks like originally. So this is a sequence that we were supposed to get, just a bunch of histidines. And then after this insertion of this A, so here's our mutated sequence, we can see that every amino acid after the point of the mutation has been altered. Same thing with the deletion. So here, same normal sequence, and then now we've left out this A. And then after this point of mutation, every single base has been altered. Now, there can also be mutations of the chromosomes that we're not talking about occurring during interphase. We're talking about occurring during meiosis. And this is called non-disjunction, where the chromosomes fail to separate and could result in a trisomy situation where you have 3 copies of a type of chromosome. So with non-disjunction, this is what we see. So we see normally how the chromosomes are going to split from each other in anaphase 1, and then the chromatids split from each other in anaphase 2. And if they don't split, we could end up with egg or sperm cells that have an extra chromosome, which would result in trisomy, or egg or sperm cells that have no chromosome, which would result in a monosomy situation. A karyotype is a chart that shows homologous chromosome pairs. You can detect gender by examining the 23rd pair of chromosomes, and we can detect non-disjunction mutations, including trisomy. You might also be able to detect insertion, duplication, mutations. So here we have a karyotype of a normal female, and we know it's a normal female because there are 46 chromosomes in the 23 pairs, and the 23rd pair are 2 X's. Here we have a normal male, so 46 chromosomes in 23 pairs, the 23rd pair, 1 X, 1 Y. So maybe we could see with this one that there's been some type of duplication that's happened here, or maybe a deletion of the chromosome, so they've lost pieces or gained pieces. This would be a situation of trisomy, so this is Down syndrome. There's 3 copies of the 21st chromosome. And with this one, this is a situation of a monosomy, to where there's only 1 X chromosome. So the 23rd pair, there should be 2 X's or 1 X and 1 Y. This one only has a single X. Then to restriction enzymes. Restriction enzymes are used in genetic modification and genetic manipulation. And they are enzymes that cut DNA at specific sites. So we saw this before we did a practice SOL question, to where we saw the sequence of CTTAAG, and the particular type of enzyme EcoR1 would cut that sequence between the A and the G, and that's the only place it would cut is in the sequence CTTAAG between the A and the G.